In this question, you have to identify the enzyme that facilitates opening of DNA helix during transcription. Option A, DNA polymerase. This is incorrect because DNA polymerase is responsible for synthesizing DNA during DNA replication and not transcription, and it does not open the DNA helix. Option B, RNA polymerase. Now, RNA polymerase indeed unwinds the DNA helix at the promoter region during transcription and synthesizes RNA from the DNA template. So, it facilitates both the opening of the DNA as well as the synthesis of RNA. Hence, it is the correct option. Option C, DNA ligase. It is also incorrect because it is involved in DNA replication and repair, where it joins the fragments of DNA such as Okazaki fragments together, but it is not involved in the unwinding of the DNA helix. Option D, DNA helicase. This is also incorrect, though DNA helicase unwinds the DNA helix, but it does so during DNA replication and not transcription.